I believe for now it's the best we have. Okay, but the question if it's ideal platform and toolkits and treatment plans, and the answer to this would be I don't think so. We still need to do a lot of work to address key shortcomings of those systems. One is really there is no ideal delivery system. You mentioned AAV, adeno-associated vectors, and lentiviral vectors. Lentiviral vectors are a very good system, but the potential of this system is the caveats of this system or shortcomings come from integration nature of lentivirus. So lentivirus integrates into genome, which can increase risk of oncogenicity, toxicity associated with integration, et cetera, et cetera, et cetera. So to tackle this problem, we actually develop and we focus our research on so-called integrase-deficient lentiviral vectors, which very similarly to adeno-associated vectors cannot integrate. So it's not integrative platform, but it has very other features, good features from lenti, such as large capacity, efficiency in packaging therapeutic cargoes, efficient delivery into dividing and non-dividing cells, such as cells in CNS, as you know, it's not dividing cells, etc. So it's really an innovative system which I believe has bright future. Adeno-associated vectors, it's a number one platform for gene therapy right now. With development of novel therapeutics based on AAV vectors, it become more and more popular. However, it has also shortcomings, such as small size. You cannot utilize bulky therapeutic cargo, or it's difficult. You have to modify the system to fit the small packaging capacity of AAV. And CRISPR-Cas, for example, is a very bulky system. If you want to create something that we call all-in-one, which comprise all the components of CRISPR-Cas, that's difficult. So that's another direction that science is taking to create elements of these innovative tools which would fit into this viral platform. Now, I'm by training virologist, and my background in development of these vectors, but I open to also to other systems which recently starting to emerge, such as nanoparticle, lipid particles, all this nano system which not really derived from there is no association with vectors. They're not there yet, but one day they will be. I can see 10 years, 20 years from now, they will take a lead in delivery methods, and they possibly could compete or even replace vector systems, because they have obvious advantages. The major shortcoming for now, they're not there yet, not efficient. They're not at the level to utilize these therapeutic tools and to deliver efficiency, especially when we're talking about gene therapy and clinical applications. But one day, I can definitely foresee the system compete with lenti, AAVs, and even could potentially replace viral-mediated gene-to-cell transfer.